Good morning. Welcome to the Interfex webinar series. My name is Kate McCarthy, and I'm the Director of Marketing and Technical Content for Interfex. Thank you for joining us this morning. Today's topic, solving the implementation challenges of continuous feedback control in cell culture processes. Just a few housekeeping items. We're recording today's event, and each of you will receive an email tomorrow with a link to view the recording. If you are currently having difficulties hearing our speaker today and are listening through your computer, please check that your speaker volume is up, or consider dialing in through the conference call line instead. The dial-in information is on the right side of your screen in the audio portion of your control panel. Feel free to ask questions throughout the event using the questions box location, which is located on the right side of your screen in the control panel. Jane will respond to your questions at the end of the presentation. And now please join me in welcoming our speaker, Jane Ring. Jane is a research scientist, Integrated Pharm Solutions Applications Development with AMD Millipore Corporation. As an application scientist, Jane has worked on various product development teams, including the Mobius Single-Use Bioreactor Program and the Cell Culture Media Development Program. Prior to joining AMD Millipore, Jane built a very strong cell biology background, performing early-stage R&D, developing implantable gene therapy delivery systems. She also produced monoclonal cell lines against target antigens. And now, please join me in welcoming Jane Ring. Jane?
Bear with us a second, ladies and gentlemen. We're just having a slight technical difficulty with the slide's ability to advance. Good morning. I'd like to apologize for the technical difficulties we're having this morning. We can't seem to advance our slides, but hopefully we've worked out that issue, and we can start. So today I'm going to present some data revolving around our Mobius Sensor-Ready technology. I'm going to offer you guys some background thinking behind the Sensor-Ready technology. I'm going to discuss some of the electronics pump design, the increased flexibility that the Sensor-Ready offers, and its ease of use. Next slide. So basically, our Sensor-Ready technology is basically an external circulation loop that's used for sampling and process monitoring. The idea behind the Sensor-Ready technology is that it offers flexibility for process monitoring and control without any custom modifications of your bioprocess container. The assembly has the ability to house anywhere from 2 to 8 probes, which really does open up the door for additional process monitoring beyond your standard pH and DO control. The Sensor-Ready assembly is compatible with your standard PG 13.5 threaded probes that are 12 millimeter in diameter and anywhere from 100 to 120 millimeters in length. They are manufacturer independent, so the customer has the choice of probes they choose to use. Next slide, please. All our probes are calibrated offline prior to installation into the Sensor-Ready assembly. And once these probes are installed securely into the assembly, they are ready to be sterilized. I think what I like the best is that this method of probe installation really does enable the user to check for proper probe insertion into the assembly prior to the whole sterilization process. And as a general guideline, we sterilize at 121 degrees for 30 minutes using a liquid cycle. And once these assemblies are sterilized, they're really easily connected to the bioprocess container using the Link sterile-to-sterile connectors. And due to the simplistic design of these connectors, there really is an increased confidence in your connection process to the assembly to the bioprocess container. Next slide, please. One of the major components of our Sensor-Ready assembly is the electronics pump that maintains the circulation of your process through the loop. Next slide, please. Electronics pumps are based on the principles of magnetic levitation. The actual pump impeller is suspended and contact-free within a sealed casing that's driven by the magnetic field of the electronics motor. The magnetic levitation creates a low shear design that results in gentle handling of your cells as they flow through the loop. Next slide, please. Just to assess the shear effects the pump has on the cells, we did some initial work in collaboration with Dr. Jeffrey Chalmers at Ohio State University to determine the shear sensitivity of CHO cells. Dr. Chalmers has developed a microfluidic flow contraction device that has become known as the torture chamber that has a 256 by 483 millimeter channel that he uses for all these evaluations. He performed analysis on 4 CHO cell lines. 2 were provided by us, and 2 had been already evaluated by Dr. Chalmers within the system. All the cells were in mid-log phase at the time of testing and used 500,000 cells per mil as the density to put through the channel. He used 4 flow rates ranging from 30 to 90 mils per minute that he has correlated to energy dissipation rates of 1 million to 100 million watts per meter cubed. Any results that we obtained from this work enabled us to define the energy dissipation rates that correlate to cell damage. Next slide, please. So Dr. Chalmers collected samples from the flow contraction device for each flow rate. He clarified them to remove any cell debris and ran them on the Promega Cytotox-1 assay to measure the LDH levels within each sample. And the higher the LDH level equated to higher cell death. The LDH levels were then used to determine the percent lysis within each sample, and then this percent lysis was correlated to specific energy dissipation rates. And you can see here is the energy dissipation rates that initiate cell lysis fall between 1 million and 10 million watts per meter cubed. Next slide, please. These cellular EDR levels were then compared to simulated RDADR levels that are found within the electronics pump that is attached to our sensor-ready assembly. Simulation studies to determine these EDR levels within the pump were run at 3 liters per minute using a fluid that had comparable density that you would find with unsupplemented chemically defined media. And the diagram on the right, on the top, shows a planar cut through the electronics pump from top to bottom. The bottom diagram is a left to right through the center of the pump through the impeller. The scale to the left shows the range of EDR levels that were found within the pump. And you can see is that most of the EDR levels that were determined to be within the pump fall well below the 300,000 watts per meter cubed. But there are a few select points within the pump that reach a maximum energy dissipation rate level of 500,000 watts per meter cubed. Now, if you take the data that was determined within the pump and you compare it back to the data that was collected by Dr. Chalmers, you can see that the energy dissipation rates found within the pump fall well below the minimum EDR level required to initiate cell damage at 1,000 watts per, 1 million watts per meter cubed. Next slide, please. So once we had the simulated pump data and cell data correlated, we wanted to set up a live test to test the electronics effect on cell growth and viability within a live system. To accomplish this, we engineered a Mobius 3-liter bioreactor to contain an external loop that contained the sensor-ready pump. The pump was set to a flow rate of 3 liters per minute, which is equivalent to the flow rate of the sensor-ready loop found within our 200-liter bioreactor system. This 3 liter per minute flow rate within the 3-liter at its working volume of 2.4 liters actually results in a recycle rate that's 80 times greater than what you would find within the 200-liter bioreactor with the same electronics pump flow rate. We harvest samples daily for a 10-day process for cell counts and viability, and then these results were compared back to a 3-liter bioreactor control that didn't have an external loop and a 200-liter bioreactor that did have the sensor-ready loop attached. Next slide, please. So as you can see here, the results from the scale-down model show equivalent growth and viability between the 200-liter bioreactor with the pump running at 3 liters per minute and the 3-liter control without an external loop. You do see a slight lag with your 3-liter with the external loop. However, if you look at day 8, all test conditions reach the same density. So this lag in growth is most likely due to the higher recycle rate that is found within the 3-liter bioreactor as opposed to electronics pump. Next slide. Next, I'd like to show you some measurement compatibility testing that we did between the sensor-ready and bulk samples. Next slide, please. So testing the measurement compatibility of the sensor-ready assembly was first done by determining the T95 conductivity profiles within the sensor-ready assembly and comparing that to profiles that we found within the bulk. For this testing, we used our 2,000-liter bioreactor at full volume, and this level was chosen for this test because it represents our most extreme configuration of our Mobius bioreactor platform. We used 2 conductivity probes. One was placed into the bulk of the tank, and the second probe was placed into the sensor-ready assembly, and then the bioreactor agitation was set to 10 watts per meter cubed. To initiate these conductivity measurements, a bolus addition of 1.25 molar sodium chloride was added to the top of the tank, and data was collected for every second until a plateau was observed. The data was then normalized, And the recorded mixing times are the point at which the probes reached 95% of their final averaged value. And as you can see here, the curves that were generated from the collected data really do show that the sensor-ready assembly is reflective of the bulk. Next slide, please. Then we did a second test to compare the process data that we collect from the sensor-ready assembly and the bulk. This was done using a CHO-S batch process at our 200-liter scale. Within this test, we had to modify a bioprocess container with a swabbable needle-free valve to allow us to sample from the bulk. Now, sampling from the sensor-ready assembly has two ways. You can either harvest through your swabbable needle-free valve or using a closed-loop tubing line that allows for sterile sampling. But due to the configuration of the bulk sampling with the swabbable needle-free valve, we chose to sample from that valve as well using the sensor-ready assembly. We did pull samples daily from each port for cell growth, viability, glucose, and dissolved gases. Next slide, please. And you can see here is that results showed comparable measurements from all the measured parameters between the sensor-ready assembly and the bulk. It is worth noting quickly that there is a minimal amount of purging that's necessary when you're sampling from the sensor-ready assembly. This is due to the configuration of the sensor-ready assembly. All these sample ports are located at the very top of the assembly that prevents the buildup of any cellular debris within your lines. Therefore, it really does minimize the amount of purging that's necessary. Next slide, please. Next, we did a case study to look at some cell culture performance using the sensor-ready loop. Next slide, please. So the sensor-ready assembly had been incorporated into a variety of cell culture processes using multiple cell lines in order to demonstrate its diversity of use. There were 4 cell lines that would run in both the 3-liter and our 50-liter Mobius bioreactors. In this instance, we chose to use the 50-liter bioreactor as our large-scale model because this one actually represents the most extreme case for this configuration because the contents of the bioreactor will cycle through the loop approximately 4 times an hour throughout the duration of the culture. And all the data then was collected from the 50-liter was then compared back to our 3-liter control. Next slide, please. So the first process was our in-house CHO-S process, which is our monoclonal antibody process. It's a 10-day batch culture with a seed density of 200,000 cells per mil. It was run at 14 watts per meter cubed at a pH of 6.95 with 30% DO with daily sampling. And as you can see here, the Mobius 50-liter bioreactor performed equally as well as our 3-liter control. Next, please. The next process was an adherent cell culture process. Here we used the human mesenchymal stem cells that were attached to solo hill collagen-coated microcarriers at 7 grams per liter. In this instance, the 50-liter bioreactor was run at the 10-liter working volume, and the 3-liter was run as the 2.4-liter working volume. All cells were seeded at 10,000 cells per mil, and they were run at 37 degrees. Within the 3-liter, the agitation was at 80 RPM, and within the 50-liter, it was at 75 RPM. And again, here you can see that we had equivalent growth between the Mobius 3-liter control and the 50-liter bioreactor with the sensor-ready assembly. You can also see on the right-hand side the photos of the cells on the microcarriers, and you can see they really did have very similar morphology on the surface of the microcarriers between the two systems. Next slide, please. So next, there was an evaluation that was performed for vaccine production, and this was done by researchers at Valneva. They used a duck embryonic stem cell-derived cell line to produce measles virus. They used a fed batch process utilizing EV66 cells that were inoculated at 400,000 cells per mil. The cells were cultured at 1/3 of the final operating volume for the first 3 days at a pH of 7.2, 50% DO, and 37 degrees C. After 3 days, the bioreactors were filled to their final operating volume of either 2.4 or 50 liters. The operating temperature was lowered to 33 degrees C, and then infection was initiated with the measles virus. The measles virus production was run for an additional 5 days. And again, here you can see comparable growth and viability, growth and virus production between the Mobius 50-liter bioreactor with the SensorReady loop and the 3-liter benchtop control. Next slide, please. And the final process outlines an insect cell process that was used to produce hepatitis C virus-like particles. These growth profiles with SF9 cells that were used for the production of this VLP were generated in a study by researchers at IVET. They also used, obviously, the 3-liter, 50-liter bioreactors. They were inoculated SF9 cells at 500,000 cells per mil. They were allowed to grow for 24 hours at 30% DO, 27 degrees at 6 watts per meter cubed. After 24 hours, they were infected with baculovirus at an MOI of 2 infectious particles per mil. And then the culture was continued for an additional 4 days, and then harvest and purification of the VLPs. And again, you can see here that viable cell densities and viabilities were similar between the 2 cultures. Next slide, please. And finally, I'd like to show a little data highlighting the flexibility that the Mobius SensorReady offers. Next slide. Because the SensorReady assembly is a modular design, it really does simplify the implementation of new sensor technologies into your system. The flexibility of being able to connect 2 assemblies in tandem creates the possibility of increased process control by creating space for both redundant pH and DO probes, as well as probes for measuring additional parameters. And best of all, you can do all this additional capability without any customization of your bioprocess container. Next slide, please. To highlight this flexibility of the SensorReady assembly, 4 additional probes were added to the assembly during a 2,000-liter bioreactor run. They used a Mobius SensorReady quad assembly that contained redundant pH and DO probes, and they connected that in tandem to a dual Mobius SensorReady assembly that contained a CO2 sensor and a viable cell density probe. And these samples were pulled daily for 11 days. And here you can see that we did have consistent measurement between the redundant probes for pH and DO, and we also had successful monitoring of the CO2 through the duration of the run. Next slide. And the viable cell density probe showed a great correlation between the capacitance units measured within the SensorReady assembly and the offline viable cell density measurements that were done on the ViCell. Next slide. So in conclusion, we've shown the ability to integrate up to 8 sensors within the Mobius SensorReady assembly really does provide ultimate flexibility for bioprocess control and monitoring without any customization of your bioprocess container. We've shown that the Labtronics low shear bearingless pump produces EDR levels that are significantly lower than the EDR levels shown to negatively impact CHO cell growth and viability. That sampling from the Mobius SensorReady assembly sample ports provides accurate measurement that's representative of the bulk with minimal purging of your lines. And we've also demonstrated the ability of the Mobius bioreactors to be run with a variety of cell types grown in either suspension or microcarrier-based cultures and their use in various applications. Next slide. I'd like to thank all the scientists and engineers from both EMD, Millipore, and our collaborators for all the great data that helped me pull this presentation together today. And now I'm ready for questions.
So thank you, Jane. We do have a couple here for you. The first one is, what happens if the tubing on the loop gets pinched or the pump stops during a run?
So as part of the sensor-ready loop, there is a non-invasive flow meter that's used in the loop that constantly monitors the flow through the loop. So if at any point the flow is reduced, an alarm gets triggered within the software of the bioreactor itself.
Okay, next question, is the pump rate adjustable?
Unfortunately, no. The rate is set within the controller software.
And then let's see, how was the 3 LPM chosen for pump rate?
So that was chosen based on a balance of considerations between moving the cells through the loop quickly in order to minimize the residence time and maintain good representation of the bulk. So while choosing the RPM that would still be comfortable for shear and with the majority of mammalian cell lines.
Okay, great. And just a reminder again, if you have any questions, you can submit them on the box on the right-hand side of your screen. We do have another one. It's 2 questions in one. What is the residence time of the cells as they pass through the loop? And are there concerns that oxygen could be depleted by the cells as they traverse the loop?
So the residence time of the cells, so the flow rate through the loop is set to 3 liters per minute. So at this speed, it takes approximately 20 seconds for the cells to cycle through that loop. And also based on the likely density of your cells that are going to be in your tank and the oxygen transfer rate of the cells, they really won't ever be hypoxic.
Okay, I don't see any other questions at this point. I do want to remind everyone that they can forward any questions that they have to Jane. And if there's anything that we have not answered, she'll respond to you directly. But I'd like to thank you for listening today. Jane, it was very informative. We appreciate you joining us today. And to learn more about the Mobius single-use bioreactor, they'll be at booth 317 at the Bioprocess National Conference in Boston on October 26th through the 29th to stop in and see them firsthand. But on behalf of Jane and myself, we'd like to thank you for listening and have a great day.